Hi everybody, if you're watching this it means that I could not make it to class. So I'm going to run through the slides the same way that I would if I was standing in front of you. The one difference is going to be that for the Pear Deck slides, obviously I won't be there to, wait for your responses and then click show response and all those things. So whenever we have a Pear Deck slide like this with a question, you guys can feel free to, pause the video when you see a question slide come up, think about the answer, and then I'm just going to move right on to explaining it. So if you want to, try to answer the questions for the Pear Deck slides, which I recommend you do, you should pause the video on your own, try to answer the question, and then, continue watching to hear the solution. First one here, what is the correct order of the phases of mitosis? So we can look here, we've got a couple that start with prophase, 3 start with prophase, 1 starts with prometaphase, so we're going to eliminate A. And then we've got one that goes from prophase to anaphase, not that one. Prophase to metaphase, not that one. So D is our correct answer. Prophase, prometaphase, metaphase, anaphase, and telophase. Remember that one that is prometaphase is, the half prophase and half metaphase. So it always goes in between those. So if you see a question like this, always look for prometaphase and make sure that it's sandwiched right in between prophase and metaphase. All right, what is pulled apart in mitosis? Hopefully you pause the video and now you've come up with your solution and you're going to answer. So what is pulled apart in mitosis? Choices, homologous chromosomes, sister chromatids, both, or neither. So what is pulled apart during mitosis is sister chromatids. So we are not separating homologous chromosomes in mitosis. We are only separating sister chromatids. So B, correct answer. Okay, so a little bit more review from what we talked about last lecture. We said last time that each human cell has 23 unique chromosomes. They're numbered 1 to 23 according to their length. So the longest one is called chromosome 1. The shortest ones are, chromosome 23. So if you see a chromosome of a different length, it would be a different number chromosome. And the chromosomes all have genes on them. There's just some examples here. These might not necessarily be true. I just made them up. But let's just say that chromosome 1 has a gene for hair color on it. We'll say that chromosome 2 in this scenario has a gene for height. And chromosome 3 has a gene for eye color. So remember, each chromosome has certain recipes that are always found on that chromosome. So kind of like if it was a set of encyclopedias, there's certain things, certain definitions and explanations that are always found in volume 1 of the encyclopedias, and then some are found in volume 2 and some are found in volume 3. So same thing, certain recipes are found on each chromosome, and that's just the chromosome that they're always going to be on. But remember, we said that each cell has 2 copies of each of those lengths of chromosomes because we're diploid. If a cell only has 1 of each length, that's a haploid cell. But our regular cells, not our gametes, just our regular cells, have 2 copies of each chromosome. You get 1 copy from your mother and 1 copy from your father. What I've shown here is that the genes that you get from your father are probably different from the genes that you get from your mother, at least the versions of them. For example, on that left chromosome, the one over here, so the long chromosome, which we said had a gene for hair color on it, 2 options might be brown hair or blonde hair. For height, maybe you get tall height or short height. And for eyes, blue eyes or maybe brown eyes. So notice that the long chromosome, it still has that hair color recipe. It's just, which version do you get? So in each of these pairs that we see here, so the red and the blue paired together, those are homologous chromosomes. They're homologous, so that first part of the word homo, that means same, because they are the same chromosome, meaning they're the same chromosome length. They have the same recipes, they just might have different versions. So, that leftmost one, those are homologous chromosomes for chromosome 1. One has brown hair as the hair recipe, one has blonde hair as the hair recipe. So homologous chromosomes are the mother version and the father version of the same, length of chromosome. Just for further clarification, so what we're looking at now is a pair of homologous chromosomes, but each of them has already undergone replication. So remember we said in class that whenever they appear as an X like this, it means that they have already replicated. So the left one there, we're going to say that's homologous chromosome, A. Maybe that came from the mother. And the one on the right, that's homologous chromosome B. Maybe that's the one that came from the father. So we'll say red from the mother, blue from the father. Now each of those, because it has already undergone replication, now it has that X shape because it's really two chromatids, two twins that are stuck together. So the one on the left here, this leftmost bit of the X, that would be one chromatid, and it's stuck together. In class, we said they were paper clipped together. So it's paper clipped together with its identical twin here that was made during replication. And the same thing here on the blue one. So this was the one from the father, but because it was replicated and it's still paper clipped to that new one that was made during replication. That's why it looks like an X, because it's two of them paper clipped together in the middle. So red is one homologous chromosome. Blue is the other homologous chromosome. And then each part of the X is a chromatid. So we've got chromatid 1 of homologous chromosome A and chromatid 2 of homologous chromosome A, and then the same here for homologous chromosome B. So make sure that you're clear on the difference between the homologous chromosomes, which are the mother versions and the father versions, and the chromatids, which are basically just the, identical twins that are paper clipped together. So you only have sister chromatids after you have undergone replication. When we did mitosis last time, we were separating the sister chromatids. The DNA had replicated, and then we were just pulling apart the two copies, pulling apart the two paper-clipped copies. Okay, this is another review question from the end of last lecture when we talked about cancer. What is the function of a mutated tumor suppressor gene? So remember, a tumor suppressor gene, not the mutated version, just the regular normal version. A tumor suppressor is a cell that is like is a gene that is like a brake pedal for the cell. So that keeps the cell from dividing if there's a problem. So maybe the DNA is damaged, or maybe the cell has physical damage to it. There's some reason where the cell should not divide. And a tumor suppressor is a normal way that the cell can hit the brakes and say, we are not going to go through the cell cycle, we are not going to divide because this cell is unhealthy. However, if you have a mutated tumor suppressor gene, that means that even though the cell is unhealthy, it is still going to divide. So A here says unable to prevent the damaged cell from dividing. A is going to be a correct answer. If this had said, what is the function of a tumor suppressor gene, not the mutated version, just tumor suppressor gene, then B would have been the correct answer. If it had been asking about what is the function of a proto-oncogene, then C would be the correct answer, because the proto-oncogene was the gas pedal when cells should divide. It's telling a healthy cell, you look great, we think you should divide. That's the proto-oncogene. And then D would be if the question had been asking about what is the function of an oncogene, which would be the gas pedal that is stuck on, and even an unhealthy cell gets told, go for it, divide. So, but this question was asking about mutated tumor suppressor, so that would be A. All right, so that was the review from last time. If you're thinking right now that there's something that you're confused about, you can try to shoot me an email. Depending on why I am not in class today, maybe I'll be able to get back to you. Or if there's another teacher that maybe is filling in a little bit, you could always ask them. So enough of review from last time. Today we are going to start talking about meiosis. So meiosis is a word similar to mitosis, but it's a different process. So mitosis, which we've already talked about, is just the regular dividing of diploid cells. So just regular old cell division. You have a cell, you want to make another version of that cell, you just directly are going to copy everything. Meiosis is a special process for making haploid cells. So our gametes, which are our sperm and our egg cells, those are haploid. So we said earlier today that haploid cells only have one copy of each chromosome. So remember we had said that in order to make a diploid offspring, we take sperm cells that are haploid, one copy of each, egg cells that are haploid, one copy of each chromosome, and then when they combine, sperm plus egg combines, then you get a diploid offspring. So in order to have these special sperm and egg cells, we have to somehow take our diploid cells and make these haploid gametes. Okay, so meiosis is the process of making these haploid cells or gametes, the sperm cells in males or the egg cells in females. A haploid cell is never going to have a homologous pair of chromosomes because they only have one copy of each chromosome. Okay, so anything that's haploid, it doesn't have a mother version and a father version in there. It's just going to have one chromosome. Chromosome 1, one version of it. Chromosome 2, one version. Chromosome 3, one version. So it does not have two copies of each chromosome. It only has one copy of each chromosome. So showing basically what I just explained, but with oranges. So if we want to have a diploid offspring, what we do is one parent, in this case the father, provides the haploid sperm, which is half, and the mother provides the haploid egg, which is half, and then we end up with the full orange, the full diploid offspring. All right, so question then, if a diploid cell of an animal has 10 total chromosome segments within it, how many unique chromosomes does it have? So think about what it means to be diploid. So think, okay, if they have 10 segments, how many of them are going to be unique chromosomes? Diploid means that a cell is going to have two copies of each chromosome. So if this animal has 10 segments, but we know it's diploid, that means that it's only going to have 5 unique chromosomes because it has two copies of each. So 5 times 2 copies of each would be the 10 chromosome segments that it has. So this would be B5. Total, there are 10 chromosomes. It's just that only 5 of them are unique. This would be similar to in our human cells, where we can say we have 23 unique chromosomes, but actually if we counted the number of chromosome segments in a cell, we have 46 chromosomes in each cell. 23 unique, but then we've got 2 copies of each. Okay, so it's good to use oranges and to think about it that way. I'm going to do some of the describing today using me and my husband and my daughter as an example, because it's always easier to think about things, if you can picture, okay, so that would be my mom, that would be my dad, if I had a kid, so we're going to try to put it in sort of a human perspective today. So let's see, so here's my husband, here's me, so he would make sperm in his testes, I would make eggs in my ovaries. The way that I would make my eggs, or that he would make his sperm, is through meiosis, which is what we're talking about today. And then of course we have sperm plus egg fertilization, we get a zygote, and you can see that in this case the egg had the blue colored chromosomes and the sperm had the red colored chromosomes, and now they have combined in this zygote here. So now we have 2 copies of each. So the sperm had 3 chromosomes, and the egg had 3. Obviously they didn't put all 46 in the cartoon picture here. But then you can see once the egg has been fertilized, now we've got 2 copies of each of those, which means it's now diploid. And then through cell division and mitosis, lots of growing, and then you end up with a baby at the end. All right, so we've said already meiosis is how we make our gametes. And it seems like what should happen is what's on the left here, which you can already see there's an X through it, but it seems like what we should do if we want to make haploid cells is that we should start with our diploid cell, our whole orange, and we would just slice it in half, and we would make one cell that has half the DNA and one other that has half the DNA. But that's not what we do. That would make perfect sense. But instead what we do is we start with our whole orange, which is our regular diploid cell. Then the DNA replicates, just like it does before mitosis. So in the S phase of interphase, we have DNA replication. So this is meant to show here that we now have, 2 oranges. It's kind of an orange with another orange by it. So the DNA replicates. Then it splits. So because it replicated and then we had 2 oranges, then it splits, so now we have 2 separate whole oranges, and then each of those gets chopped in half. So we end up with 4 half oranges. I guess this one kind of looks like a quarter of an orange, but each of these is meant to be a picture of a half orange. So instead of doing the easy thing and just taking a diploid cell and dividing the DNA in there into 2 haploid cells, first we replicate the DNA, we split it once, and then we split it again. So the result of meiosis, instead of just being 2 haploid cells, the result is that we make 4 haploid cells. Replicate the DNA, split it once, split it twice, and then we end up with 4 haploid cells. And now I've added some chromosomes onto my orange. So we can see here, in this case, we have just 2 unique chromosomes. We've got long chromosome and we have short chromosome. So we've got long red and we've got long blue, and then we have short red and we have short blue. And so this is, again, what doesn't happen. It seems like we should just split it in half, and each one gets 1 copy of each chromosome. This got a copy of the long chromosome and a copy of the short, long and short, but again, that is not what happens. Instead, this is our same orange up here, but now we can see replication happened. Now they look like X's. Remember, anytime that the chromosomes look like X's, it means because the DNA is replicated and the 2 chromatids are paper clipped together in the middle at the centromere of the chromosome. They're stuck together. And then we can see that we do 1 split, and so now we have a cell, or an orange, a cell in this case, that has 1 copy of the long chromosome, 1 copy of the shorter chromosome, and the same on the right. 1 copy of the long one, 1 copy of the short one. Then, the second time it splits, we end up with the X's pulled apart, kind of like in mitosis, okay? So this long blue X got separated, and so now one of these oranges here, one of these cells, one of these gametes, ended up with 1 bit of that large blue X, and this is where the other large blue X chromatid ended up. The same thing, that little red one got split apart into singles. So you can see how we started up here like this, diploid, 2 copies of the long, 2 copies of the short. And each of these ones that we have down here, each of these half oranges, each of these haploid gametes that we made, is going to have 1 copy of long and 1 copy of short. You'll notice some of them have red as the long, some of them have blue as the long, some of them have blue as the short one, and some have red as the short one. But they all only have 1 copy of each length of chromosome. Meiosis has 2 parts to it. Meiosis 1 is where we excuse me. Meiosis 1 is where we separate homologous chromosomes. So the mother versions and the father versions get separated. And if we look back at our previous slide here, that means we're talking about this first split here. And we can see that of the long chromosome, we had the mother version and the father version. The blue big chromosome went one way, the red big chromosome went the other way. The same thing with the little short chromosome. One short chromosome went one way and the other short chromosome went the other way. That means that we split the homologous chromosomes. So this one on the left has one version of this long chromosome, just the blue version. And this side of the long chromosome, we only have the red version, okay, and opposite for the short one. Just happens to be opposite. I could have put 2 blue here, I could have put 2 red here. The point is that the long chromosomes get split up, the short chromosomes get split up. That's meiosis 1. Meiosis 2 is where we separate the sister chromatids. That's the same thing we did in mitosis. That's where we pull apart the X's so that the paper-clipped, conjoined twins get separated. So in meiosis 2, we could see we had these X's here, and then when we go from this row to this row, the actual X's themselves get pulled apart so they're just straight lines. So these sister chromatids get separated. And here's a nice slide. If the oranges were confusing you, some people really like the oranges because it's helpful to see when you have a whole and when you have a half. If the oranges were confusing to you, this slide is a little bit more, scientific looking. So we can see at the top here, we started with a cell. This is after the replication already occurred. But we've got one big red, one big blue, one little red, one little blue. We separate the mother and father versions of each chromosome. So if we're looking at the big chromosome, we want to make sure that the red big goes in one direction and the blue big goes in the other. So we just happen to have the big blue went left, the big red went right, and for the small ones, we want to make sure we separate that mother and father version. So we want to make the small red go one way and the small blue go the other way. So that's how we separate homologous chromosomes. At this point, each cell should only have one chromosome of each length, right? Because that's what the homologous pairs were. There was the 2 versions of each length. Okay, so the left cell here has 1 chromosome of the long length and 1 chromosome of the short length. Yes, there's 2 chromatids that are attached, but there's only 1 version. We only have blue version here, we only have red version here, and for the little one, we only have the red version here and the blue version here. Okay, for meiosis 2, this is where we separate the sister chromatids. So that means that we're pulling apart the 2 halves of an X. So we can see that we've just pulled apart those 2 bits. So the left one, that big blue X, got pulled apart. The little red X got pulled apart, and on the right, the big red X got pulled apart and the little blue X got pulled apart. So you should be able to look through this figure and clearly identify, okay, I can see how we've separated homologous chromosomes. Okay, I can see that, we still have chromatids attached together, and that once we get down here, that those chromatids have separated now. All right, which of these cells could be the result of meiosis 1? So think about what happened in meiosis 1. We said that we wanted to separate the homologous chromosomes. So the mother and father versions had to be separated. I know these are all in green, but it shouldn't matter. You should be able to answer the question not based on color necessarily, but you can answer the question based on length. So we said that the homologous chromosomes were the 2 versions of a chromosome of the same length. So if we want to separate those during meiosis 1, that means that the result of meiosis 1 should be a cell that only has 1 copy of each length So A, both these look like big X's. So those big ones did not get separated. In B, they're single strands, they're single chromatids, but they both look long to me. So again, they did not get separated. D, we can see that we clearly have 2 of the long chromosomes and 2 of the short ones, so that didn't separate them. However, C down here, we only have 1 version of long and 1 version of short. So C could be the result of meiosis 1. Homologous chromosomes have been separated. Okay, this figure looks complex, but we're going to kind of walk through it. So this is the whole process of meiosis. We looked at a similar one of these when we talked about mitosis before. So we can see, sorry, this is my husband's computer. Anyways, where was I? So we can see the different phases here. And remember, DNA is always replicated in S phase. So we have interphase here, and we can see that we have single squiggles, and then what happens between interphase and prophase 1 is now they're X's. In this picture, it's all crammed into one slide, so you can't see it too well. We are going to look at these in more detail, but the main part to see here is just that we've got 2 parts of meiosis, meiosis 1 and then meiosis 2. Okay, each of the meiosis has the same phases that we saw in mitosis, so these names should look familiar: prophase, prometaphase, metaphase, anaphase, telophase. And all the ones part of meiosis 1 have 1s after them, so prophase 1, metaphase 1, you know. And the same thing here for meiosis 2. Prophase, prometaphase, metaphase, anaphase, telophase, and they all have little 2s after them. So we have meiosis 1, we go through all the phases. Meiosis 2, we go through all the phases. Don't forget, I'm going to say it 50 different times in this video, meiosis 1 separates homologous chromosomes. Meiosis 2 separates sister chromatids. Okay, and if you kind of want to pause the video and walk through these phases on your own, you can kind of see how it's hard to see the length of the X's, but you can get an idea of, okay, I can see that they've separated the ones that are longer, they've separated the ones that are shorter, and then over here in meiosis 2 we started out with X's, and the X's get pulled apart. You can see those chromatids getting pulled apart. Meiosis 2 should look pretty similar to mitosis, in terms of you have the X's that line up in the middle, and then the X's get split apart into just single strands. And then at the end, cytokinesis. So we started with our 1 cell, and then at the end of meiosis 2 we have 2 cells, and then those get split, and then at the end we have 4 cells. Okay. One thing that is going to be different between mitosis and meiosis is something that happens early in prophase 1. The homologous chromosomes are going to form something called a synapse, which is shown in the picture here. So when they come together, the mother version and the father version of a chromosome, and they've already been replicated, which is why they look like X's, they're going to come together, they're going to form a synapse, and when they're in this formation, they're also called a tetrad. So TET is what am I trying to say? The word that means 4. So a tetrad would be because there are 4 sister chromatids. Okay, so like a tetramer is going to be something that has 4. So tetrad means that we have 4 chromatids. 2 pairs of chromatids, but there's 4 if you count them all. So, blue number 1, red number 1, blue number 2, red number 2. We've got 4 chromatids. So sometimes you'll see this called a tetrad when they line up together like this. And the reason that they have to line up is because there's a process that they undergo called crossing over or recombination. Again, this is only something that happens in meiosis prophase 1. So this is a specialty thing that happens in meiosis. And so what happens is the mother and the father versions of a chromosome, so the homologous chromosomes, they are going to swap some of their DNA. So before looking at the letters here, I'll just point out, let's say that the blue one is the one that came from dad, the red one is the one that came from mom, and we can see that they're, they line up next to each other, they're real close, they're sort of intertwined. The result down here is that you end up with a chromosome that is mostly blue, but it has a little tidbit of red, okay? And then you'll see here, this chromosome is red, but it has a little tidbit of blue. So we end up with these chromosomes that have, swapped some of their DNA. So originally, we were saying the blue one is the one from dad, but after they do recombination, this is now, mostly from dad, but has a little bit of mom. So we end up with these chromosomes that are formed that are not just the mother version or not just the father version. It can be a single chromosome that has some bits that came from mom and some bits that came from dad. And this is really helpful, which we're going to see as we go through a few more slides. This is helpful because when you make a haploid gamete, when you make a sperm cell or when you make an egg cell, you only pass along one copy of each chromosome. Okay, but in your body, you have one chromosome, if we just talk about chromosome 1, you have one copy of chromosome 1 that you got from your father. You've got one copy of chromosome 1 that you got from your mother. But when you're making an egg cell or a sperm cell, you can only pass along one copy of chromosome 1. So, how do you choose? How does the body choose? Which one do you pass along? Do you pass along chromosome 1 from the mother, from your mother, or chromosome 1 from your father? Because we have crossing over recombination, there doesn't have to be that either or. You could pass along chromosome 1 that is sort of this mix of some of your father version and some of your mother version. So you're still passing along just one copy of chromosome 1, but it can be sort of this mix and match, chimeric version that has both bits from your father chromosome 1 and your mother chromosome 1. Okay, anyways, back to the slide. So if we look at the letters, the dad version here had all capitals as for its genes. The mother version in red here had all lowercase, and then we can see that not only if we look at the actual color, but if we look at the letters, that means that now this chromatid here is going to have, capital A, capital B, but it has the little version of C. If we just gave the straight father version, it would have capital A, capital B, capital C. But now we've got the version of C that can get passed along is the mother version. Okay. So, if we look at some examples, let's say that this is your long chromosome. And let's just say that the genes on it are hair color gene and feet size gene. Okay, and the version from mom, we'll say the red version, is has the brown hair recipe and big feet. And the one from dad was a blonde hair recipe and a little feet recipe. Okay, so what I had said before is that when you are making your sperm or egg, you can only pass along one copy of this one. Okay, so that would mean that you're either passing along brown hair with big feet or blonde hair with little feet, meaning that you could never pass along to your child brown hair with little feet or blonde hair with big feet. Only one or the other could get passed along because they're, stuck together on one of those chromosomes. However, this is where our recombination comes in. So recombination can swap the DNA so that you can pass a single chromosome that is more of a combo, a combo of your parents, which would be the grandparents of your child. Okay? So, for example, the before crossing over options are you can make an egg cell or a sperm cell with brown and big. You could make another one with brown and big, because remember we make 4 during meiosis. We could do blonde and little or blonde and little. So you would either be passing along brown hair with big feet or blonde hair with little feet. You could never swap. Okay, the after crossing over or after recombination version here is that now we could possibly still pass along brown with big, if this one didn't, oops, sorry about that, if this one didn't recombine, we would have brown with big, and if this one didn't recombine, this one on the far right here, then we could still pass along blonde with little. But now we could also pass along the option of brown hair with little feet or blonde hair with big feet, okay? So there's 4 different versions here of that chromosome that can be passed along in a gamete. Okay, so before crossing over, you could only make egg cells or sperm cells with these, traits together. After crossing over, we could now have a sperm or an egg cell that has, these different combinations. Okay, so if we go back to, real people, so Harriet's mom, Harriet's dad, here's Harriet, and these would be her body cells. I'm not putting all 46 chromosomes in because it's too much. We're just going to stick with 2 chromosomes, long version and short, and then because these are her body cells, she's got 2 copies of each. So of the long chromosome, oops, sorry, I keep clicking. The long chromosome, she's got 1 from dad, and then she's got 1 from mom. And of the short chromosome, she's got 1 from dad and 1 from mom. So before she makes her gametes, this is what her cells look like. That's what we just said. Those are her diploid body cells. After the replication occurs, they look like X's now, all right? After recombination, then they would look like this, okay? So now she's got, of the 4 chromatids now, she's got 1 that is still, all dad. She's got 1 that's dad with a little bit of mom. She's got 1 that's mom with a little bit of dad. And then she's got 1 that's still all mom. And this happens for every, length of chromosome. So here, again, we just show, long and short. But imagine this is chromosome 1, this is chromosome 2. In her actual cells, she would have chromosome 3, 4, 5, all the way to 23. And each of those lengths of chromosomes undergoes this recombination, this swapping. Down here, I came up with this word, maybe it will be helpful or not. When they're sort of the mother and father versions together, I sometimes call them combosomes. They're chromosomes, but they're a combination of the mother version and the father version, so a combosome. But that's not an official scientific word. It's just something that I do pepper in through some of these slides because I think it's easy to understand that we're talking about one that has mother version bits and father version bits together. So, when there's the blue and the red stuck together, it's a combosome. It's a chromosome that has some from each parent. Okay, so now we said that after recombination, that's what the cell looked like here. Okay, so that's where we're picking up at. That's the same cell. I just copied it over to this slide. So this is what happens after recombination. And now we're going to talk about meiosis. So when Harriet makes her egg cells, each haploid egg cell is going to get 1 copy of each. So they all have to have 1 copy of the long chromosome and 1 copy of the short chromosome. Okay? But they can get either of them as long as they get 1 long and 1 short. So in the previous slide, we saw that of her long chromosome, there were 4 different chromatids. 1 was all dad, 1 was mostly dad with a little mom, 1 was all mom, and 1 was, a little bit of mom and a little bit of dad. For the long and for the short, okay? And when Harriet makes gametes, each one needs 1 copy of each length of chromosome, but they can be mixed and matched in any way. So for example, she could make an egg cell that has chromosome 1 that's all dad, and maybe the chromosome 2, the short chromosome, is this one that's, partially mom, partially dad. Or that one that was all dad could get paired with this one, or with this one, or with this one. So is it possible that, that she could make an egg cell that happens to be pretty much only the stuff she got from her father? Sure, it's possible. It's not likely. It's more likely that her egg cells are going to be a representation of both her father and me, instead of just one or the other, meaning that her future children would be a combination of my husband and me in the chromosomes that Harriet has provided. So picture-wise, kind of mapped that out. So Harriet's egg cell likely is going to have, long chromosome that has bits of her dad and bits of her mom, me. The same thing with the little chromosome. But don't forget, okay, I had said that, it's likely that her egg cells are going to have a combination of me and her father, in the egg cells that she provides. But, my grandchildren, Harriet's future children, they are not going to just be a combination of us. Me and my husband, that's what Harriet provides, but, she is going to, her egg cell would have to meet with a sperm cell, in order to make that diploid offspring. So don't forget that, Harriet's children, so that would be my grandchildren, they're only going to be at most half that's Harriet, or half that's my husband and me. That's the parts that Harriet provided to her child, and the other half is going to be whatever her, future Prince Charming sperm has provided. Okay, so think about that. And then I've just labeled these as blue here, the ones that come from the sperm of the future. But, each of these, you could put little pictures on saying that, these would be the other grandparents. So this would be the sperm cell of Harriet's future Prince Charming, but we could put little pictures of Prince Charming's mother and father on each of these, which would mean that, Harriet's child, which would be my grandchild, has chromosomes that are, some of them are a combination of me and my husband, and some of them are going to be a combination of whoever the parents of Prince Charming are. So it's kind of interesting to think about that when you have a child, and when your child makes their gametes, that the DNA of you and the DNA of the other person, husband, wife, partner, whoever, is going to actually be linked together, permanently in the future. So recombination is basically the process of biologically sticking together bits of the two parents' DNA. So right now in Harriet's cells, all of my DNA is separate from all of my husband's DNA. But when Harriet's egg cells are made, that means that some of his DNA and some of my DNA are going to become, attached together. And there's now going to become chromosomes that are produced where the two of us are permanently attached together. So, kind of interesting to think about. And obviously, I'm using myself and my child as an example here, but, feel free to reverse and kind of work through all these slides and think about you in this scenario, or think about your parents, as being the people in this scenario. And it's interesting because it really can give a kind of an insight into why certain people might really, really look like, their paternal grandfather, or maybe they really, really look like their maternal grandmother, and you think, wow, it's almost like that other parent isn't really represented at all. And part of it is because the DNA that gets passed along, depending on how the gametes are formed, there might be different combinations. Like what if this had been the chromatid that got passed along? That means everything on chromosome 1 would all be representative of my husband's chromosome 1. Whereas if this one was the one that was passed along in the egg, means everything on chromosome 1 would be representative of me. So it really just depends, when the gametes are made, how these things get sorted out. And it also depends on how the recombination occurred. I have here, this looks about half of it's blue and half of it's red. Recombination could occur where we get some red, some blue, some red, some blue, like different patterns like that, we could end up with a tiny, tiny, tiny blue at the top and everything else is red. So it's not always, a 50-50 recombination. All sorts of different versions can be made through recombination. Okay, so let's see some reminders about crossing over. The examples that we're showing only have 2 chromosomes, but remember, this whole process of recombination is happening for all 23 pairs. By the time that this is all in prophase of meiosis 1, way at the beginning. So by the time we're actually doing, splitting up the homologous chromosomes, we're not really splitting up the mother version from the father version, because each of them are these, combosome versions. So it's important when we initially start describing meiosis that we, say, okay, we're separating the homologous chromosomes, so we're separating the mother version and we're separating the father version. But once we've talked about recombination, we can see that we don't really have just the straight red version or the straight blue version. They're each more of these sort of reddish, sort of bluish, some bits from the mother, some bits from the father versions of chromosomes. But it still is important to know that the homologous chromosomes are separating. It's just that when they separate, it's not, mother version here, father version here. They're sort of these combos of mother and father. And as I sort of mentioned before, this is important because it increases genetic variation. We can pass along traits on the same chromosome even if they came from different parents. So review that slide where we had the hair color and the foot size, where we said brown hair could only be passed with, I don't remember if it was big feet or little feet, and blonde hair could only be passed with whichever the other one was, big feet or little feet. But because of recombination, now we could pass, the brown hair with the little feet and the blonde hair with the big feet, and all those different combinations. So recombination is important because it increases genetic variation. Important to remember, though, only one version of each gene is included on a combosome. So you're still only passing along one copy of hair color, for example, either brown or blonde. So even though chromosome 1 that gets passed along in a gamete is going to be a combination of recipes that came from the mother and recipes from the father, there still is only one copy of each recipe. So if your father gave you brown hair and your mother gave you blonde hair, you can still only give one of those in each gamete. You could make a gamete that has brown hair and a different gamete. We could make an egg cell that has brown hair. You could make a different egg cell that has blonde hair. But you're not going to make one egg cell that has the brown hair from your father and the blonde hair from your mother. So each gamete still only gets one version of each gene, which means that when you make a gamete, an egg cell or a sperm cell, half of your recipes get included and half of them don't get included, right? So for your hair color, if you had brown and blonde, and for your foot color, your foot size, if you had big foot or little foot, only one of each of those recipes gets included and half of them don't get included. So keep that in mind. Don't think that recombination means you can pass all the recipes that you got from your father and all the recipes that you got from your mother. No, you're still only making a haploid gamete, which gets one copy of each chromosome. And that copy has one copy, of each of the genes on it. So this is just pointing out, the other version is not passed along. So we said that, I guess this is a new pretend example. Chromosome 1, let's say now that it has eye color, ear shape, and toe hair. So when Harriet makes an egg, it would have either my eye color or my husband's, my big ears or my husband's little ears, my bald toes or my husband's hairy toes. It can't have, both. But because we make these combosomes, we could make you know, we could pass along blue eyes, small ears, and hairy toes, or we could pass along hazel eyes, big ears, bald toes, or we could do, you know, basically you have to just pick one of each recipe. One of these two, let's say blue. One of the ear recipe, let's say big. One of the hair recipes, let's say hairy toes. So that would be blue, big, and hairy all together. You just have to make sure you get one copy of each recipe. Making these combosomes just means that, you know, you could, for each recipe, it's sort of a this one or this one, this one or this one. And it doesn't matter if you get the eye color from, you know, the mother, you could still get the hairy toes from the father, depending on how recombination, you know, allows it to occur. Okay. I'm not going to read all the text on here, but because this is a video, you know, if the way I'm explaining it isn't helpful, you could, you know, kind of pause and reread all the text on your own at your own pace. Question. So crossing over occurs between options, sister chromatids, B, homologous chromosomes. The answer is B, homologous chromosomes. The sister chromatids are going to be the two identical twin paper-clipped together versions of the same homologous chromosome. So when we cross over, we're swapping red bits and blue bits. Okay, those are the homologous chromosomes. The sister chromatids are going to be, you know, the two blue bits, or not bits, of the two blue chromatids attached together, or the two red ones attached together. So we're not going to swap between the two blue because they're identical twins anyways. And we don't swap between the two red ones because the two red ones are identical twins anyways. What we do swap between are the blue ones and the red ones. So between the homologous chromosomes, not between the chromatids. Another way that we get genetic variation is what happens during metaphase 1. Okay, so remember in meiosis 1 we're separating the homologous chromosomes. In metaphase 1 is where we would see the two big versions separated. So here on the top we can see we have the big long chromosome, and then we have this one that's the shorter one. And we can see up here, the blue one got put over here, and the red one got put over here. And each of these after meiosis 1, we end up with, they kind of skip to only show metaphase because it's easier to show them lined up in the middle. Anyways, this would be during meiosis 1, and then this is what they look like during meiosis 2. So now we can see that we've got one version of the long chromosome, it happens to be blue, one version of the long chromosome over here, it happens to be red, and the same thing down here, the red version and the blue version. Because we've already talked about recombination, it would have been a better cartoon if they showed this one is blue with little red bits and this one is red with little blue bits, but they're keeping it simple and just pretending that this is all the blue one and this is all the red one. But we know that, you know, at this point recombination has occurred, so they shouldn't just be blue and they shouldn't just be red. They should be sort of that mix of red and blue. But anyways, back to what's happening here. The top here showed that the blue versions of each went to the left and the red versions went to the right. And then down here they just show what happens when the final gametes are made. So after we do meiosis 2, we pull the parts of the X apart so we end up with the straight lines. Okay, so this is one option. On the bottom, this is just showing that this is also equally likely to occur. When they arranged up here, at the top here, we saw that it was the blues were on the left and the reds were on the right. But what can happen is you could end up with blue on the left and red on the right, or you could have blue on the right and red on the left. It doesn't matter that left-right order of the mother version and the father version. So here we can see that the result is that because the long chromosome arranged itself blue on the left, red on the right, the blue of the big one went to the left, and the red of the big one went to the right. And that for the shorter chromosome here, red happened to align on the left, and so it went left, and blue happened to align on the right, so it went right. And then we can see the gametes that would result. So now we have big blue paired with little red, and these two ended up being big red paired with little blue. Each of these is equally as likely. So for each pair of homologous chromosomes, and remember there's 23, this is just showing 2 here, but for all 23 homologous pairs of chromosomes, whether the father version goes left and the mother version goes right, or whether the whether the mother version goes left and the father version goes right, is just 50-50. It's like each time these homologous pairs get together, these guys flip a coin and say who wants to go left, who wants to go right. These guys flip a coin, who's going to go left, who's going to go right. So you can end up with these gametes that are, more likely to have father chromosomes or more likely to have mother chromosomes. Because we know that they are combosomes, it's just that, maybe the blue one here is mostly blue with a little bit of red, and maybe the red one here is mostly red with a little bit of blue. So you end up getting this mix and match of, the long chromosome might be mostly dad, or the next one might be mostly mom. So genetic variation also occurs just by how they happen to arrange when they're actually splitting up. So the arrangement in metaphase 1 is random for each pair. As long as you split up the 2 big ones, as long as you split up the 2 medium ones, and et cetera, et cetera, et cetera, for the other pairs of chromosomes, whether father goes left and mother goes right, or vice versa, is all okay. I'm not going to spend too much time on this slide. We talk mostly about humans in this class, but this is just to point out that, we are usually, we're diploid, animals in general, diploid, and then we make these haploid gametes if we're, sexual reproducers, and then they combine, and then we're, the diploid individual again. Well, other life forms sometimes do it differently. So here showing fungi in a life cycle where they are pretty much always haploid, except then they go through this, short diploid phase. So everything on the bottom here in purple, this is all haploid, and then for a short time they go through this diploid phase before they split up again during meiosis and then have this haploid phase. It's not too important to understand. It's just kind of interesting to point out that there are some life forms that mostly live in this haploid phase, and then they just temporarily become diploid, and then they become haploid again. Same thing happens in plants. Again, I'm not going to walk through the whole cycle here, but in plants they have a similar thing. Sometimes they're going to be haploid, and sometimes they're going to be diploid. Those of you that are interested in plants, I would suggest, reading through the text on here. It's kind of interesting if you think about it. The benefits of, reproducing, with this haploid and diploid phase is that you can increase your genetic variation, because anytime you have sperm and egg, these haploid gametes that meet together, you're mixing, genetic material from 2 completely different individuals. And so it's always helpful to have some type of life cycle where you can do sexual reproduction. Things that only do asexual reproduction, it's more difficult for them to get a lot of genetic variation. They have to sort of do a few other tricks in order to get there. Okay, so we're almost done. What we have here is a nice comparison of meiosis and mitosis. It's important that you understand the differences, especially for taking a test. So, so far last lecture we talked about mitosis. Today we talked about meiosis. I know a lot of today was kind of background about recombination and really driving home the difference between homologous chromosomes and sister chromatids. And we didn't actually mention the phases too much of meiosis. That's because they're just really similar to what we already went over during mitosis. So I'm going to walk through this slide which is a great review slide and a good comparison. So we have in mitosis and meiosis, we're going to start here in, we're going to start the explanation in interphase. And remember that DNA is replicated in the S phase of interphase, all right? And now meiosis up here, we've got prophase 1, which is where that recombination event occurred, okay? Then we go to prometaphase, metaphase 1. So now the homologous chromosomes, the pairs of homologous chromosomes are lining up in the middle. And then here we see they're getting pulled apart. So the two big X's are pulled apart, the two little X's are pulled apart. Okay, and then telophase 1, you know, they form their separate cells. So now we have two cells, and then what's going to happen in meiosis 2 is we're going to pull apart the sister chromatids. Now that's the same thing that we're doing in mitosis, which is why they have sort of extended this arrow and they've lined up meiosis 2 with mitosis, because those are the similar processes where we're pulling apart the two halves of an X. So we're taking out that paper clip at the centromere and we're pulling apart those chromosomes. And again, we see those phases we recognize: prophase, prometaphase, metaphase, anaphase, telophase. In metaphase, this is where the X's line up along the plate, and then you would pull apart the two halves of the X. The outcomes are different. Okay, so from meiosis, we start with that one cell, we end up with 4 haploid cells. Mitosis, we start with a cell, we end up with 2 versions of that cell. So we start with 1 diploid, we end up with 2 diploid. Down here, this chart is pointing out some of the, the important processes and when they occur. So we have the first one here, DNA synthesis, and if we look, downward, that occurs in S phase for both, for meiosis and mitosis. Next process, synapsis of homologous chromosomes. So this is where they formed that synapse, that tetrad formation, where the homologous chromosomes, cuddle up together. It does not occur in mitosis. It occurs during prophase 1 of meiosis. Crossing over, or recombination, again, does not occur in mitosis, but it's during prophase 1 of meiosis. Homologous chromosomes lining up during metaphase 1 of meiosis does not occur in mitosis, right? Because in mitosis, you're only separating sister chromatids. So in mitosis, the X's individually line up, and then they get pulled apart. The two parts of the X get pulled apart. So in mitosis, we never have where we have 2 X's lining up, sort of in pairs, with their buddy, and then the buddies get pulled apart. Mitosis, we only see the paper-clipped halves of the X's get pulled apart from each other. Let's see. And then here, sister chromatids line up. That happens during both. Okay, it happens during meiosis 2 in meiosis, and it happens during metaphase in mitosis. Remember, in mitosis, we don't give them numbers of 1 or 2 because it's just one type. It's just regular old prophase. You don't have to say prophase 1 or 2 because in mitosis, there's just one prophase. So that's a good hint or a good clue. If you ever see a Roman numeral after one of the phases, it's talking about meiosis, because the ones in mitosis, they don't distinguish between 1 or 2. Okay, last thing here, they list the outcome. Okay, so what you get at the end. Meiosis, you get 4 haploid cells. Mitosis, you get 2 diploid cells. Again, I suggest that you kind of walk through each of these. You could try to zoom in, use the figures in your book, and make sure that you're kind of clear on what separates at each point. If I were to say the sister chromatids are lined up on the metaphase plate, and you had to say, does this happen in meiosis 1? Does this happen in meiosis 2? Does this happen in mitosis? You'd have to think about, okay, when do the sister chromatids, line up there, getting ready to get pulled apart? Well, they get ready to get pulled apart in meiosis 2, and they get pulled apart in mitosis. They do not line up in meiosis 1, because meiosis 1 splits the homologous chromosomes. Here's some differences that I wrote a little easier to follow format. So mitosis ends with 2 diploid cells. The new cells are identical to the original. There's no recombination. There's just 1 round of division, and this is in regular cells. Meiosis, you get 4 haploid cells. The new cells are all going to be different from the original. Remember, the original cell was a diploid cell. All of these are going to be haploid. Recombination does occur in meiosis. There's 2 rounds of division, and this is only if we're making gametes. Okay, that is my final slide. So I hope that this was helpful. Again, let me, or if I'm really unavailable, somebody else know if you have any questions.